There we go.
Got it. Um, all right. 15 people here. 1 minute over. I think we can get started. Um, Hi everyone. I'm Keoni Gandall. I I guess I'm the speaker of this talk, and right now I'm 21 years old. I've been doing synthetic biology and genetic engineering for about 8 or 9 years now. Um, in a variety of places. Have had a home lab for a number of years, kind of on and off, doing active work in it. Um, 4 years at UCI, working on directed evolution and mitochondrial engineering, and about 3 years now at a little over 3 years at Stanford, running the Free Genes project, which is a project to make genes free and open source. And during that time, I've been very involved in the DIY bio and biohacker um, communities. Uh, and so I know personally most of the people who head labs in at least the United States for community biology, and a lot of the kind of gray projects that go on in the community. Um, and also, I'd like this to be a pretty open talk. I am pretty anarchist, and so I don't believe in hierarchies. And so if anyone wants to just jump in and ask me questions, please feel free to. Um, yeah, so I guess I'll start off with how I got into biology back in the day. And then we can go towards what we're doing now, and I'd like to leave most of the time for random questions. Um, When I was in 6th grade, there was a virology textbook at the local church book fair, and I saw this college textbook on viruses, and it was really exciting to me. As the 6th grader, I was like, what are viruses? How do they work? What is going on here? So I bought this book for like $2 or $3, brought it home, started the first chapter, and had no idea what was going on. You know, they were talking about like bacterial cell walls, and I was in 6th grade. But I kept at it, and you know, after about a month or so, I was able to figure out what was going on in the first chapter. Um, and after that point, my was kind of sparks, and I was really interested in biology. And so over the summer of 6th to 7th grade, I took an AP Bio course online, um, did all the molecular biology portions, and disregarded the rest. Um, and that was super fun, super enjoyable memories from that. Um, in 7th grade, my 7th grade teacher realized that I was super into bio. And the reason is that we had a test on, oxphos and all that. And instead of just saying, how much ATP comes out of, like, the Krebs cycle and oxidative phosphorylation and stuff, I drew out everything, and then also had the full Krebs cycle with all the different intermediates, and then drew out a chemical structure of ATP on that back of the test. And he's like, "Wow, you're a big nerd." Um, and in response to me being a big nerd, as a good teacher would be, he's like, "Hey, you want to do some bacterial engineering at home? You can order a kit to my classroom, and then you can do it at home." And I was like, "That sounds awesome." So in 7th grade, I genetically engineered my first bacteria to glow green, and that was pretty wild back then. It was like, wow, I can make life, I can change life. Um, and so I continued building up my home lab with, instead of getting an Xbox for Christmas, I would ask for like an incubator. All of those I still have, by the way, which is great. And I'd go on eBay and buy PCR machines and stuff. And in 8th grade, I decided to take on a new project for the science fair. I wanted to engineer Halobacterium, this salt-living archaea. I wanted to engineer it to be green. So it's like same experiment I did in 7th grade, except applied to a new organism, kind of like a hello world in a new language. And I did that all at home, and I went to the science fair. in that science fair, I had actually eventually got up to nationals and this Broadcom Masters, and won one of the first place best prizes in the National Science Fair, which gave me, I think, $4,500 to help build out my lab. But I also got to beat Obama, which was cool. At the regional fair for that project, though, a graduate student in the cardiovascular labs actually like judged my project and was like, "Holy crap, this is like really advanced for you know, we do cloning and PCR in our labs, and it's kind of hard, and like you're doing it at home and you're in like 8th grade, like what the heck." So he invited me to come work in their lab over the summer because they had this summer program, and they were a cardiovascular lab. And so around that period of time, I was figuring out what my kind of career ambitions would be. And I go and talk to the professor, and the professor says, you know, we're a cardiovascular lab. What do you want to do? And I told him, like, very directly, is like, I want to build a synthetic cell. I want to build a synthetic genome. And the professor's like, you're in the wrong lab. You should go to this lab. And so kind of weirdly through their summer program, I went to this different lab, but since there wasn't all the formal mechanisms in place after it ended, I never left. So I just kept working there at the Lou lab for the next couple of years, you know, strengthening my biotech skills. Spent more time at UCI than at high school, which probably had a big effect on my grades. So that was all fine and dandy. And so I was doing a lot of genetic engineering at lab, getting real lab skills, and then also doing it at home, still on this quest to build a synthetic genome. You know, it was 8th grade when I really wanted to start building the synthetic genome. And so over the period of years, I thought a lot about how to do that. And I was really inspired by kind of hacker culture and the history of computer science and the Free Software Foundation and Linux and all that stuff. And so I got to thinking about how are we going to do this with cells? How are we going to apply the same principles of openness that we've learned from the computer age and apply it forward to what it will be in the coming future. And so at the first Biohacker conference, Biohack the Planet, I decided to give a last-minute presentation. That presentation was, it was about at 6 o'clock on this Sunday night, and at about like 3 o'clock or something, I saw the slot was open for, you know, 3 hours ahead, and I was like, hey, Josiah, who's a buddy I knew, it's like, hey, can I take that spot? And he's like, yeah, sure, whatever, go ahead, yeah, whatever. So I whip up a presentation, kind of bring together my theories of how we should be building things, and I give this talk. It was about how to make a buildable genome, and there were 3 principles that I brought up. In order to build a buildable genome, we're going to need 3 things. We're going to need, first of all, get really good at putting cells together, or putting DNA together. So lowering the price to actually build DNA and to test things out. The second thing we're going to need is a ton of collaboration, an open source toolkit to engineer life, engineer biology, because the amount of people working on synthetic cells right now, there's just not enough people. We just need more people to be interested in working on building life. And the final thing is we need software to tie all these experiments together so that you can easily, you know, pass results between different people. And all of that was inspired from Linux. Turns out the talk right after me was Drew Endy, who is a professor at Stanford that you guys probably know of. And it turns out the things that he wanted to talk about was this idea of the bionet, a network of biological objects passing between people and through networks. which was actually pretty similar to the idea that I was talking about, which was an open source DNA collections that we could easily share with people to get more people involved in engineering cells. Turns out another project he had at the time that was actively getting grant funding was this thing called Build a Cell, which was a project to build synthetic cells. And so I hit a lot of what he was doing. And so afterwards, he came up to me and he's like, hey, that was awesome. You should come work in my lab. And I was like, I want to. So that year, I also applied to get into Stanford as an undergraduate. And I did not get in, darn it. And, I told Drew, it's like, I can't work in your lab because, I didn't get into Stanford. I only got into ASU got rejected from 13 of the 14 colleges I applied to. My grades were terrible. And Drew's like, who had written me a recommendation letter for Stanford, was just like, that's bullshit. Come work for me anyway. So that's how I got started on Free Genes, which I've been working on the last 3 years and kind of refining my skills, which is a project to make free and open source DNA. So what's been consuming me right now has been, there's a lot of DNA out in the world. There's a lot of synthetic DNA that is reused a lot of times in order to build things. For example, iGEM, there's this power law distribution of parts and like the number of times they've been used. So, the most popular part is like more than twice as popular as the next most popular, and so on and so forth. If you take the, the high tail end, I guess not really tail end, but the beginning of that distribution, of the things that are most used, it actually comes out to a small number of things. And so what I've been working on is figuring out what should we synthesize in order to get most of what people are doing, and then freeing that so that anybody can get that kit. Anybody can get a toolkit to engineer any organism, and it will come with the majority of things that you need to engineer that organism. And so that's kind of been my job in the last few years. I've had a few other ideas of cool things to do, distributing DNA in spores, and probably the idea that I'm thinking about most right now is software tools for engineering biology. The primary one is how to build a system which can inductively reason about biology. So basically, I want to automate away genetic designers. And that's going along pretty well. So yeah, that is me. Are there any questions? And I can talk about like lots of different topics around biohacking, the people involved in biohacking, what's going down in different communities and synthetic biology communities, local dramas. But I would like to hear if anyone's interested in any particular topic.
I have a question. I was wondering if you could speak a little bit about any possible applications you see of AI towards this field of creating life and cells and genomes.
Yeah, so good question. Then I'll go into Caitlin's in the answer box. For AI, there was a really good essay called The Bitter Lesson. And basically, over the last 70 years of AI, you see that the general methods which use computation to get better have been far more efficient in everything than methods that are that use humans to reason about and to make better machines based off of how humans reason about things. And we're going to see a similar thing in biology, I think. If you take general methods that can be automated, that will beat out, really smart people in labs. And that's really scary because you're going to, we're already seeing it with platforms like Ginkgo, but there are going to be people who own those, and there will be orders of magnitude better than what the best engineers can do. And so we're going to start to get in a regime in the next 10 years or so where there are people and there are companies particularly which are exponentially better at engineering life than anyone else, including these university institutions. And if they can get to the point in which they can do biological experiments better to induce knowledge about the world, you can basically replace most of what graduate students do, which is a little bit scary. When it comes to AI and synthetic biology, the best, and so I'm making analogies to AI because I think the most useful thing about AI is that we can learn from for synthetic biology is the lessons from learning about AI applied to biology, because I think they're similar fields of where we are discovering something that already exists. In one, it's intelligence, in the other, it's the biological world. And the lessons are pretty, I think, more analogous than, say, the building of a field such as computer science. So I think history is like, if I had to answer, I'd say history is the most useful thing about AI. The other useful parts would be in figuring out, doing basically design. How much does this gene express, for example, is a really complicated problem, but I think one that they'll need AI to solve. What kit would you recommend? The Odin has a $30 genetic E. coli genetic engineering kit. I would recommend against their CRISPR kits, but like just a basic GFP kit from the Odin will get you going fine. If you want somewhere more official, Carolina Biologics and Biorad have 2 good kits, but they don't ship to homes, so it's harder to get from them. But they're both very good. Those are the 2 that I started out with. What are your thoughts on the DIY COVID vaccines, especially in regards to some of the stuff Josiah and George Church are involved with? Good question. There were actually people who I won't name, but were doing the exact things those 2 were doing, months before when COVID-19 just started. They've actually administered it to themselves. We don't really know if it works, but there are lots of people doing that aren't publicizing it as much. My thoughts are, I don't really have many thoughts on the immediacy of what they're doing, because I don't really care what Josiah does. But what I'm thinking about is the future of what it implies, or what it can mean, rather. They are using RNA vaccines or DNA vaccines for this. And the interesting thing about DNA vaccines is that they're so simple. They can be literally distributed as plasmids. And you can even imagine synthesizing 200 plasmids, which isn't that expensive, to cover nearly every pathogen a human can have, making those all in a standard DNA vaccine. And so we could see the emergence of not just anti-vaxxers, but maybe rogue vaxxers, who, once the methods that Josiah and George Church are using are established enough off DNA and RNA vaccines, that you have people wildly vaccinating themselves without, government approval or FDA approval. I find that really a really exciting possibility. The other thing that the DIY vaccines are bringing up is the ability to express RNA or DNA inside of human cells in a living person, which is going to be interesting, because now you can genetically modify yourself if those vaccines really work, which I'm excited for. Have you ever experimented on yourself? And if you do not, do you plan to in the future? So back in the day, probably 2 years ago, or maybe even up to 3 years ago, I was actually planning on doing some stuff with Josiah, the follistatin genes, which enlarge your muscles. I was mostly interested in it from an engineering perspective. And I was thinking seriously about that, but it didn't really appeal to me. But what has appealed to me is not just the COVID-19, DIY vaccination. That's pretty cool, but I don't really care about the DIY part. What I would like to be able to do is take vaccines and basically extract my own blood, sequence the white blood cells in my blood, and then figure out what antibodies go to what, and then publish those antibodies for free. If you can figure out how to express a ton of antibodies, which is basically a commercial problem, you solve every single pathogenic disease known to man. It's kind of incredible, the power of antibodies. And they don't have an immune reaction in human beings to human antibodies. So if you can figure out how to express antibodies well, you can solve most pathogenic diseases. And if you can make an open source library of a bunch of people just, taking vaccines and then like extracting their blood and putting sequences out there, you can imagine a time in which, COVID-19 comes around. We identify the first person with antibodies, immediately sequence it, and then start mass production of antibodies at breweries, where you like siphon into tobacco plants and get enough to heal tons of people. It's a really exciting future. And so if I was going to experiment on myself, it would definitely be getting vaccines, extracting antibody sequences, and giving those away. Insight on how to start up your own home lab. There is one key thing, two key things, but one key thing I would say is more important than everything else. Buy what you need to do your experiment and don't go anything else. A Hello World experiment is nice and cute, but have an experiment in mind, have something that you want to do in mind, and then go build a lab in order to do that one thing. Don't try and assemble lab equipment and then find a problem to solve. That you're not going to get far with that. It's going to become boring and you're going to waste a lot of money. That is like 10x all the other recommendations. The other thing I would recommend is avoiding basically being lazy. Know that you will have to pay a lot for things and take the route of least resistance. If you want to genetically engineer something, like express a protein, don't clone that protein in your home lab, because then you have to get everything necessary to clone proteins. Go order it. It's going to cost you way more. You're going to calculate it to cost you way more. In the end, it's not going to cost you way more, because there's going to be a lot of small things that you've forgotten about, that, like the Sanger sequencing costs, or, oh, I need more polymerase, or, oh, my electrophoresis failed. Oh, it's going to take a week to get the supplies. There's a lot of stuff that will pile up. And so you really, really, if you're doing a lab experiment, just do the minimum amount. And, I've burned myself here as well, where I try and do the cheaper route and it just doesn't turn out well. Just pay what's need to pay and do what you want to do. Any other questions? I guess I could ramble some stories.
I'm interested to hear, like, what are some of your other interests besides building a genome?
So I play StarCraft a lot. If anyone plays StarCraft, you should friend me. I also do a lot of software engineering. So for example, one of the things I'm working on right now is a command line tool with some other folks to engineer biology. And one of the things that I've kind of been working a lot on is the algorithms to search DNA, to store DNA. Turns out they really kind of suck. They've been optimized for the wrong things, in my opinion, because, from what I think about biology is that we should be taking a holistic viewpoint, not just, looking at individual genomes and stuff, but looking at all genomes and looking at all the data. And there are structures of data that you can use that get very good performance for that kind of stuff. And so I'm building out like a DNA storage library that is faster, less memory intensive, less disk intensive, less RAM intensive than any other algorithm so far, off of a couple sacrifices, mainly in construction time. So I do lots of programming. I have lots of servers set up here and there and everywhere. I like to do DevOps of actual like deploying services and doing software. I'm also a huge fan of Linux. And so I ran all exclusively free and open source software except for things like, except for communication tools like Zoom or my phone. Algorithms. So there's an algorithm called, or more of a data structure called a suffix array. And the basic idea is that you can take a string like banana and so like B-A-N, yada, yada, yada. And all those characters are indexed. So like 0, 1, 2, 3. Now, if you sort those, each one of those indices by their alphabetical order, and so like the first would be banana, and then you'd have anana and nanana, and like each suffix. If you take each one of those and organize them in alphabetical order, you get this thing called a suffix array. And you're like, okay, why do I care? Well, if you want to search anything on that string, it becomes a binary search problem, since you would just take your suffix array and then you would have like your search string, you'd go halfway, and if it matches, well, then it matches. But if it doesn't, you can search if it's lexicographically more or less. And then if it's like more or lexicographically greater than that middle string, you would jump upwards and then test there. And then you can basically do a binary search to put search algorithms into logarithmic times. So that's interesting, but that's not the only interesting thing about it. You can make this thing called an LCP array, which I won't go much into, but basically it's the amount of characters shared by each one of these suffixes going down. But you can use that with the number of occurrences. You can basically count the longest common prefix in any given string because their LCP will be the largest and they'll be like right next to each other. Turns out if you string a bunch of DNA together, like all of life, 10 terabases, this is not BWA. I think that BWA is a overly complicated algorithm for a very specific reason. BWA is very good at locally compressing things, but locally compressing things is not helpful when you're looking at all of life. It actually, I think, detracts a lot from the better method of deduplication. DNA has a lot of duplication in it, and BWA can optimize, but it only optimizes locally. And so you get compression of these chunks, but why compress two of these chunks when you could just index them and put them together and then like have a separate thing that says this goes here, and then you basically deduplicate the data. Suffix arrays with LCPs make it very easy to deduplicate data. So then I made an algorithm that calculates the storage cost of a deduplication event, and then it will recurse down a given suffix array until it's basically too costly to do this compression based off the LCP and suffix array. And so you can basically get the optimal deduplication compression amount for any given suffix array. That has been really exciting to implement. And it gives you a lot better compression than BWA and FM indexes. Also, suffix arrays are, they basically, BWA and stuff basically use, or the BWA FM index algorithms basically use suffix arrays to get their speedy lookups. And so I'm just directly using the algorithm. Sorry for going on that tangent, but yeah. Any other questions about the world of biohacking and DIY biology and community labs? Sorry. What is it like working in Drew's lab? Wild. Drew is a very unique individual who thinks about things very differently than other professors, much more on a meta level, on a societal level, which I think is very useful. Should we spend a whole class period talking about Jeffersonian democracy and bioengineering? Like Drew would say, like, yes, and I'd be inclined to agree with him. So in Drew's lab, kind of these big ideas is much more acceptable to chase after than small incremental increases. And viewing how biology is going as analogous to other historical events, and seeing biotechnology as like this blip in society as a whole is quite informative for how the field will develop, which is a very interesting perspective to have on your work. It's a very global perspective. Any other questions?
It looks like we had a question a moment ago that was, if you could create your own startup in Synbio, what would it be? Not to steal your ideas.
Yeah, no, definitely. I love when people steal my ideas because then it makes them happen. There are a couple different ones. The primary one is that I believe that the way we do biology right now is not the right way to do biology. There can be, and there will be, systems which can induce and deduce information about the biological world, designs of the biological world. And the most interesting things about these systems is that they will leverage automation, which scales much better than human beings. You're going to get into a time in which those systems are orders of magnitude more effective than people. And if it is true that there is a large market for biology, those companies will make billions because they will be better than everybody else. They will basically be the Microsofts of the biological age. And so Ginkgo has been working very, very tightly on that route, and investors seem to believe in them. I think that one of the most compelling startups in the synthetic biology space would be designing a system which is able to not only autonomously run experiments, but one that is able to figure out what experiments to run to reach objective goals. Think of biology labs as computers, and we're just doing an optimization, something like that. I think that will be massively profitable, and is a very meaningful thing to contribute to. And most important key there is that it will not be solved by working on any of these like biological engineering projects. It will be solved working on the system to biologically engineer things, which is what I've kind of tried to work on my whole career. What are some regulations you think should be put on biohackers? None. I think that they have autonomy of their bodies and should be able to be, so long as they aren't infringing on other people's autonomy, should be able to do what they want. We have yet to see evidence that biohackers or DIY biologists or community lab members are a significant source of bio insecurity. On the flip side, we have seen many times in which state actors have acted, very badly when it comes to biological weapons and biological containment. And so who am I more concerned with? A bunch of biohackers or our state level governments? It's definitely the governments. Finally, I think that regulations or the fear of biohacking, the fear of community bio and DIY biology is something that was not intended to be created, but is created by large corporations in order to control the narrative and control the development around biotechnology. Not intentionally, but that is what practically it's been led, what has happened. If you look at people who say are anti-GMO, almost always you'll find that they're not actually anti-GMO. They're actually anti-large corporations owning food and owning seeds. One reason why, Europe is so much more anti-GMO than America is. Even some of the largest detractors of GMOs, they're not actually mad at GMOs. They're mad at, for say, colonialism of large companies coming into their local areas and forcing them, not giving them choice or liberties or optionality around what food they grow because of convenience or price. So what I believe is that by empowering people to make their own decisions and have their own options around what food they grow, by giving them the power of biotechnology, you actually reduce the amount of fear in the system, and thus would reduce the amount of necessary regulation. And by doing that, you also decrease the amount of power that large corporations have in order to control both the narrative and the intellectual property of biotechnology and the regulatory, and weaken their grasp on regulatory capture. So to put it in another way, I think that there should be more biohacking rather than less, because the idea that we should fear it or that there's a security threat is basically for me right now, a fable made up by large corporations to keep you under their thumb. What are some examples of cases when state actors were irresponsible with bioengineering and biological weapons? Yeah, so one experiment was in the Soviet Union. They discovered how to make smallpox far more lethal than it already is by inducing, I think, brain hemorrhaging, something like that. Another example of when the United States decided to try like dropping mosquitoes on their own people in order to figure out if how things spread, or releasing a bacillus strain into the subways of New York in order to figure out how a bacillus anthracis strain could spread. Turns out some people were allergic to that, and that causes problems. There is a good argument that SARS-CoV-2 could have come from a Chinese laboratory. I'd probably put it at like a 70% chance that it didn't, but there's some good circumstantial evidence based off how China reacted and how the virus spread that would, could have come from their facility. There are other cases in the United States of it's not just the mosquitoes, but I think there was an example of fleas getting released. Yeah, and a few more examples from the Soviet Union that I can't, put my finger on right now. But generally, state actors or academics have been responsible historically for much more of the security threats. Hey, Keoni, we met once at iGEM in 2014-15. Wow, that's a long time ago. I was curious, what do you think about the intersection between iGEM and biohacking? Oh boy. iGEM is ironic, hypocritical, maybe, in that, Randy Retberg talks about openness and not being greedy with biotech, yet continually increase the fees for iGEM year after year, which is why you see very few community labs actually participating in iGEM because they can't afford it. Why do academics have a lower price for iGEM than community labs who don't have money? iGEM itself is a competition. iGEM is an institution in which the motivation for people to participate is opportunity, unlike companies and stuff where it's money. But that opportunity is for those who can pay for it. iGEM wishes to be the Apple, whatever that means, of biotech competitions. They wish to be on the top and be heralded as the most useful and best competition, at expense of the smaller players who can't afford to participate. And there are perspectives on whether or not that's a good thing. For example, being considered the best is great if you are a small lab in a third world nation which needs to get funding, and so you can say like, hey, we're doing this awesome thing, and for iGEM to be worth it. But iGEM generally has a massive disconnect with the biohacking community and DIY bio community. I've seen it more often that iGEM, when interacting with the biohacking community, is actually a place for people with money and no real academic connections to participate. And that's what I actually think their community lab tracks are intended for, not for genuine contributions to the community lab atmosphere, community lab space, community labs community, biohacking community. They're overall quite hostile to that. Their values of openness also are, I think I've said enough about iGEM. Yeah. How can people get involved with the biohacking DIY bio community? That depends what you mean by involved. You can debate people on Facebook all day if you go on the biohacking Facebook group. You can call yourself a biohacker if you decide that you want to grind your coffee beans differently. Getting involved is self-selecting. And so, what I would encourage if you want to get involved, just go do things. Just like go do exciting, awesome, beautiful, wonderful things. And then share them once you do them, not just hype them up. So people can get involved by doing things. You can also get involved in some of the forums. There's the DIY Bio Google groups, which has slowed down over the years. Used to be much more active in 2013, 12, and 14 era. And then also there's the biohacking Facebook group, which is kind of like Josiah's little club. But it's like, how do you get involved with the biohacking community? Well, how do you get involved with the hacking community? It's like you hack, you do hacking. There's also several conferences that are good to go to, such as the Bio Summit and Biohack the Planet. Do you recommend reaching out to people/companies working on similar things for help? Yeah, of course. Networking is the most important thing there is. It's great when I'm able to reach out to a subject expert on nearly anything within the community lab space and be able to get a response because I've talked to them before. Like I know these people personally. Like if I have a question about the history of GenSpace, I can just go text a GenSpace founder. And I find that most people are more than willing to help. Most people want to be good and they want to help other people out. And so, yeah, go ahead and text whoever you want. And not just whoever you want, text more people than what you want. Ask for more help than you would think would be maybe appropriate. And I think you'll get some positive responses. Just be a good person and don't ask for too much. But yeah, most people are friendly. I also find I am a high openness type of person, and so I also believe secrets are bad. And you know, most people, a lot of people in the DIY bio community also share kind of the idea that secrets are bad, and usually are more than willing to help. Any other questions from any of our 18 participants who haven't asked anything yet? I love to talk about things and hear myself speak, but I would want it to be appropriate for what everyone would want to hear. Well, self-plug, if you want to see something more about biohacking, there's a documentary on Netflix that is quite good, where they interview, you know, Josiah and David Ishii and whole crew, and you can think it's called Biohacking. Netflix documentary would recommend. What's your best drama story from the biohacking community? Oh God. Oh God. This one's pretty great. Huge thing. So there's this guy, Andres Strummer. He is a Austrian biohacker. And if you know anything about Europe, you can't biohack there. So he normally has to get Americans to do it, his American friends. I've worked with him on a few projects. But he had a new idea for a herpes vaccine, and possibly an HIV vaccine from this kind of expired patent. And so he had approached some people in the United States, this time in Jacksonville, to get started with this. And you get this very eccentric crowd of folk, you know, conglomerating around Andreas's idea, with their own kind of histories. And they decide to go forward on this HIV vaccine. It's actually just an antibody that they express. They found an anti-HIV antibody that works, and they're like, they want to engineer someone with it, inject it. So we have this crew of people. We have Andreas, who is this wild, mad scientist who's the nicest person ever from Austria, who loves American things. We have Tristan, who is an basically an HIV survivor, who wants to benefit the community through self-experimentation of these kind of risky substances because the institutions have traditionally left them out. We have Gabe, who is very cynical, like a very in-your-face aggressive guy. And we have Aaron, who is a businessman. They don't really know where he came from, but he kind of had the money behind it. And so this group of people build a lab in Jacksonville, Florida, and they start doing things. And so then at Synbio Beta, while they were doing this, Josiah has the brilliant idea of starting to get drunk on stage. And so, you know, you have all of these guys in suits talking about their biotech pharma companies, and you have like Josiah in like a t-shirt, like with a flask in his pocket, drinking whiskey while giving a bio presentation. And he was giving it on one of his new CRISPR kits. And someone's like, hey, you should inject yourself with the CRISPR. And they were live streaming, and Josiah's like, fuck you, I'm going to do it. And so he basically gets a syringe filled with like, you know, DNA that have CRISPR plasmids to basically make his muscles bigger. And he like injects himself on stage with it. And that got a ton of media attention because they're like, this crazy drunk guy just injected himself with CRISPR on a stage. So now everybody's hyped up about this. And Aaron is like, the media will eat this up. And so they have their HIV thing working. They give a talk at a biohacking conference in Texas. And Aaron, without any warning for anyone on his team, he didn't really tell them that it was happening. He just gets up on stage, he pulls down his pants, and he just slaps his leg up on a chair, and then injects himself straight into the leg with this experimental HIV or herpes vaccine or whatever. And that gets a ton of media attention. And then he starts being like, we want to sell this as an alternative to FDA stuff, as a cure. But all the people on his team was like, we don't have any scientific data for this. So they get in a huge fight with Aaron. They start yelling at each other. Aaron takes the keys from the lab in Jacksonville, locking them out of their own lab. They have to get like the police involved. Big blow up thing. So all of the actual lab members quit for that project, and Aaron goes on his own way trying to assemble a new team. A couple months later, Aaron is found dead within a sensory deprivation chamber, probably overdosed on some type of drug that he took for that. And that is the end of the chronicles of HIV vaccine. There was a company, but I forgot the name of the company. But that was a clusterfuck all the way through. Just a wild time. So don't inject yourself with CRISPR on a stage, kids. Or do if you want media attention.
I think that's a good place to end because we're getting really close to the end of the time frame that we blocked out for this.
Cool.
Thank you very much for talking to us, Keoni.
Thank you for all your good questions. And yeah, thank you very much.